Biochemistry is great on the first day, huh? Wait till Wednesday, huh? So, welcome to Biochemistry 450/550. My name is Kevin Ahern, and I will be your instructor du jour. Very happy to have you guys here, and I have to say the class is off to an unusual start. I have never turn the microphone on. You can hear me. Can you hear me now? I've never had the class start off with silence like that. I don't know. This is very unusual. There's just, I mean, maybe I'm thinking I got my pants unzipped or something like that. I don't know. That's a sure way to get everybody's attention. Let's see. Let me say a little bit about the class, a little bit about myself, a little bit about you, and then we'll dive into some biochemistry. So I see some familiar faces. I see many smiling faces, which is always good to see on the first day of class. And I'm very happy to see that. I know biochemistry, for a lot of you, is a subject that's scary, that's intimidating, that is whatever, and I don't think it needs to be. I think the more afraid you are of something, the less likely you are to do well with it. And I really don't want you to have that fear. I don't want you to have that dread. I want you to come to this class and learn in this class. And I think the best way to do that is if you're comfortable with the class, with me, etc. So I promise that I will do with you whatever I can to make you comfortable, to help you in any way that I can. I keep a lot of office hours, and I always say, and it's true, that you can't come to see me too often. So if you have questions that you can't get resolved, that you can't understand, or the TA can't help you with, or whatever, then I'm more than happy to help you. So please come and see me. That's something that I like to do. It's a big class. This class is the biggest 400 level class taught on the OSU campus, and it's the biggest by about almost twofold. Yeah, we're number one, right? There are obviously disadvantages to large classes, and I personally like large classes. Part of it is I'm a ham, I guess, so I like to get up in front of a lot of people and make a fool of myself. But part of it is that I sort of like that energy that comes from a big class. The downside, of course, is there's only one of me, and there's a total, as of this morning, 280 of you. And that's a lot of people. And so it's partly for that reason that I videotape my lectures. I provide all the materials, online streaming, etc. The eCampus students who are at other locations can see the videos and watch and take the class as well. I feel in general that you should come to class. I think it's better for you to come to class. eCampus students, of course, can't do that, and I make myself available to them by phone and other things as necessary, but I think you should take advantage of the fact that you're here and come to class. I can't make you come to class, and I won't make you come to class, but I can also tell you the data points are that the students that I get to know and the students who come to class, I can tell the difference in the grades. There's just no kidding yourself. You can sit and say, okay, I'm going to watch those videos, okay, and those videos are good, but that's not the full deal. So you really need to take advantage of whatever is available to you. I'll be telling you more about the class, obviously, as we get going through. And if you have comments or questions at any time, I encourage you to let me know and so forth. So I'm kind of curious, just sort of a shout back at me sort of thing. What kind, when I say biochemistry, what are the thoughts that come to mind? Molecules, protein folding, enzymes, anything besides the actual components of biochemistry? Yeah. Pharmacy, okay, there you go. How many, okay, it says, thank you for reminding me. How many pre-pharmacy, pre-dentistry, pre-veterinary, pre-med, whoa, I knew I'd get outnumbered on that one. And how many from something besides those areas? Whoa, okay, so there's quite a mix. Obviously, the students that take this are almost exclusively into life sciences. You don't take biochemistry unless you have something that is there for that. This is a class that most students take because they have to, not because they want to. And I recognize that as well. So I want to help you to learn biochemistry. I want you to enjoy biochemistry as much as I enjoy biochemistry. And I think that's, and I'll have some fun things for you to do during the term, and we'll do that. The class includes recitations, and each person in the class is required to go to a recitation. Attendance in recitation is 5% of your grade. So expect that you'll go to recitation and that you will participate. Now, there's no assignments. You don't turn anything in, but we pay to have TAs in recitation to help you solve problems. And you'll see that especially this term, working through problems and so forth is a very big component. And so it's for that reason that we want you to go to recitation so that you'll have the leg up that you need to work those problems. Okay? I'm available as well to help you when you have difficulties and so forth with that. But the recitations are there for you to get the assistance that you need. And no, I do not allow you to not go to recitation. So you need to go to recitation. Okay. When you log into the class, welcome. How you guys doing? That's okay. I'm lost too, but then I'm in the middle of a lecture. So how you doing? Okay? Okay, I guess. All right. So, so anyway, I'm very happy that you're here, and I'm off to what will be, I think, an exciting term. The syllabus is required reading in the course. So you download the syllabus off the web page. I don't provide it here. I don't see a reason to waste paper. But you do need to require it. I generally ask one question off the syllabus on the first exam. So it's important that you know what's in the syllabus and you understand the rules of the course, which are in that syllabus, and you can download that right there. Most of you, when you come in, will come in. Oh, by the way, so I've got a calendar. I should also mention this. My calendar, if you click here, is available. And my calendar shows where I am, what I am, etc. I have two offices. I'm working in the Honors College now, and I'm also working in biochemistry. So my two offices are in ALS 2145. That's my biochemistry office. And I have an office in the Honors College, which is Strand Ag Hall 229. How do you know when I'm in each one? If you look on here, for example, you'll see tomorrow morning at 8 a.m. I have UHC. UHC stands for University Honors College. And when you see that, I will generally be in that office for 90 minutes. So from 8 o'clock until about 9:30, I'll be over there. You can see my schedule of meetings and so forth. And that tells you where I am. You do not need to make an appointment to see me. And I don't even post specific office hours. My policy is when I'm in my office, you're welcome to come anytime. So on average, I'm in my office about 25 hours a week, depending upon the office. If you want to make an appointment and make sure you know where I am, I'm happy to do that as well. But you do not have to make an appointment to come see me. And if for some reason you can't find me at any of my office hours, then let me know and I'll make other arrangements. I've been known to meet students on evenings and weekends if they were unable to do things. So I know you have jobs and things like that. Sometimes it's difficult to make those connections. So my calendar will always tell you where I am, when I am, etc. And my class always has the highest priority. So I want you to have priority in terms of things I need to do. So when I'm meeting with somebody, like I am with Alex in the morning, come by and let me know that you're there, and I will finish and then quickly get to work with you. Okay? So that's my plan. Let's see, what else? So that's my calendar. My calendar is linked there. I'm on Facebook. Come see me on Facebook. I enjoy making a lot. In fact, I made several friends in here already out of Facebook, which is kind of cool. The main thing you're going to go on this page for is to find this. Okay? So this is the class schedule page. It tells you a little bit of technical things about the pages. I videotape each lecture, as I said, and within about 24 hours, it varies a little bit from time to time, depending upon how busy I am and how busy the campus server is. Within about 24 hours, I will post on here links to streaming and downloadable versions of this video. You can put those on your iPod. You can watch them over the internet. You can do whatever you want to with those. I make podcasts available for those that you can also pull down. And I make what I call highlights available. Highlights are my written summary of what I thought was the most important things that I talked about in the lecture. Now, I give you all these materials, and it's very easy to look at that and say, oh my God, there's just going to be so much material. And there is a lot of material in the course. But these are all things that I'm providing to you to help you to learn. Okay? I'm not giving you extra things to pile on and say, okay, now here's another chapter that you can go read and you'll be responsible for on the exam. Okay? My policy on exams is that I can assure you that I will never ask you a question about something I haven't talked about in class. So when we look at information in the class, the highest priority of information is what I talk about in class. Okay? The other things in the class are there to help you better understand what I talk about in the class. So my highlights are my summaries of what I thought the highlights were about what I talked about in class. Okay? The textbook is there to help you, again, see that information in a different way from what I've been talking about and help you to better understand it. The textbook, of course, has problem sets and things like that that can help you to understand the material better. Okay, as you probably have guessed, I am a pacer, and I'm told that if you watch my videos on high speed, it's a great tennis match. I go back and forth and back and forth and back and forth and back and forth and back and forth, okay? If you go on fast speed, if you go on normal speed, it sounds like I'm talking at fast speed because I talk too fast. And I don't mind you raising your hand and saying, hey, can you slow down? A lot of times I get going and have no idea how fast I'm going, okay? So if that happens, then I will definitely try to slow down and work within those time constraints. Questions, comments? This is the quietest group I've ever seen. Except for that person over there going, yeah. All right, so there are, of course, the other reason to come to class is there are, of course, occasionally technical problems. We have a technical problem with the camera. We have a technical problem with the audio or something like that. And, of course, if you were counting on that to be there and I don't have a recording of it, then you're going to be lost. So make sure, of course, that you get as much as you can out of this course. Okay, that said, if there are no comments or questions, we're going to start our journey through biochemistry here. Yes? Good question. I just had the TA this morning take over 2 books in the library. It takes the library a little while to get them there. I'm sorry, to get them posted. I'm sorry, they're not probably there today. They should be there, I would think, in the next few days in the library. So yes, I do put those books in the library for you to check out. And I will tell you, I wrote a textbook a few years ago, and my sympathy is with students at the ridiculous prices that the companies charge for textbooks. And I can tell you it's not due to the royalties that they pay authors. It's absolutely not the case. But the textbook prices are outrageous. If there's things I can do to help you around that, I will do that, of course, but I do have a textbook for the course, and so you'll find it. As I said, it's a supplement. I'm not going to ask you things I don't talk about. So things I talk about are the highest priority. Yes? It's right at the top of the thing. Oh, okay. So it's OregonState.edu/instruction. Okay. And /BB450. So if you go to that page, right there, /BB450, that'll get you to this page, and then everything else links from there. Okay? Okay, so this is a class for undergraduates and graduate students. Common questions I get are, well, are the graduate student grades going to affect our grades? And the answer is no. The curve is set by the undergraduates, and so the distribution of grades and so forth that I post are based on the undergraduate grades. The graduate grades are superimposed on top of those. So the graduate grades do not affect the distribution for the undergraduates. Okay? Other questions, comments? My tie on straight? Yes, ma'am. No, you don't need the book in class. Absolutely not. No, no. Another hand? I thought I saw a hand. Maybe that was the same question. Yes? Are there vastly different requirements for graduates and undergraduates? Graduate exams are, they take the same exams. Graduate exams are graded more rigorously than undergraduate exams. Graduate students absolutely have to know how to spell. Undergraduates have to know how to spell pretty well. Okay? Graduate students, if you can't spell, and I had a graduate student last year that was a wonderful young man, and he was wonderful at biochemistry, but his spelling was absolutely atrocious, and it killed him on a couple of the exams. So if you haven't gotten to the point of being a graduate student and you can't spell, then you're going to need to work on that. Okay? Yes, and I will say this, by the end of the term, he was spelling much better. So. Recitation will meet this week. So yes, if you're in recitation tomorrow, you will be expected to attend recitation tomorrow. Yeah. Clear as mud? Oh, yeah. Songs? I don't know anything about songs. It's possible. It's possible. There's a rumor to that effect. Okay. What's that? Oh, yeah. what do you bring to recitation? I would bring a piece of paper to write things down on. I will tell you what I tell the TAs. I had a TA meeting this morning. What I tell the TAs is that the first thing that the TAs are supposed to do in recitation is say, are there any questions? So you bring your questions to recitation. You bring a piece of paper to write down the things that you might learn in the problem solving and get those taken care of. So your questions are the first things that the TAs are supposed to work on, and then they have things that they'll talk about with respect to problems. If you ask questions for the whole period, that's fine too. But your questions and getting your needs taken care of are the first priority of the recitation. Okay, yeah, thanks for asking. Yes, back there. TAs will have office hours, and I'll have those posted on the schedule page. If you scroll down this page right here, what you will see is it says TA information to be announced. And they've actually given me their office hours and everything. I should have that posted probably this evening sometime, and you can find out when to meet with them. Keep in mind, and by the way, some of you times I get requests for tutoring and so forth, and I'm happy to help you line up with a tutor. But I will tell you that TAs are free tutors during their office hours. Okay? So don't be afraid to go and ask them for assistance as if they were a tutor during their office hours, because that's what I'm paying them to do. Okay? If you need additional help or you want some other outside tutor, then of course I'm happy to help you get that as well. And I've got some excellent students who do tutoring in biochemistry. And if that happens for you, then of course let me know. Other questions? It's a good group. Should I call roll now? Yeah. Do I do any research at OSU? No, I don't. Actually, I teach and advise students. I got out of the research business about 20 years ago because I decided that what I really loved was teaching and advising students. And I feel extraordinarily lucky to be able to do what I do. And so I like that. That's why I like these first days of class. I like the interactions a lot. And so I don't do research. And I don't miss it. I tell you, the most wonderful thing that OSU has is sitting in the seats right out here in front of me. And that's pretty cool. So that shut everybody up. Wow, power here. Okay, well, let's dig in. If I don't see any other hands, I'm looking upstairs, I'm looking downstairs. Okay, have open minds. Your open minds are going to be filled with biochemistry. Now, as I said in the email I sent out to everybody last night, if you didn't get the email, then maybe you're not listed on the list on the Blackboard list very well. By the way, I don't use Blackboard, so you won't find, if you go looking for the course on Blackboard, it won't be there. You need to go to the website I gave you here to find the class. I recommend in general downloading. I've got all the figures for the class available. They're both available in PowerPoint and PDF formats, same files, and the outline. If you download that and you bring it in here, then you're not trying to quickly scribble down all of the notes or all the figures and so forth that I've given you. So you can write on the figures if you bring them to class with you. I think that is the best way to go. I see somebody over here who has already done that, and I strongly recommend that. I think it's the best way to take notes. Okay? If you go to buy the textbook and they provide all these things for the instructor's notes and all that, or the textbook notes and so forth to write on, you can have that as well, okay? But I'm providing this to you. If you have used textbooks, you're basically going to see the same figures that they're going to provide in there. So, I just noticed also one of my TAs is here. That's Jessica Morgan, who is sitting on the side. Jessica will be a TA for 2 of the recitation sections, and you will get to know her. Do I have any other TAs here? I don't see any of my other TAs. They're not required to come. Okay, well, let's talk about biochemistry. When I say biochemistry, most people have the notion in their minds, well, we're talking about the chemistry of living cells. And the chemistry of living cells was where biochemistry had its roots. Okay? The idea that cells had chemistry going on inside of them is something we've realized for a long time. And for a very long time, it was felt that that chemistry that goes on inside of living cells is really exotic. It's really very different than what's happening in the rest of the universe. Okay? And over time, people came to realize, and by over time, by the 1800s, people came to realize that that chemistry that was going on in living cells was no more exotic than the chemistry that was going on everywhere else. There are things that happen in living cells that don't happen anywhere else, but at the level of chemistry, that chemistry is, in fact, exactly the same. Okay? So that's the roots of biochemistry. The real modern roots of biochemistry date to about the 1930s. And in the 1930s, a man named Schrodinger had the rather radical idea that And by the way, I'm just giving you general information. You're welcome to take these notes if you want, but you don't have to. I will also tell you one other thing, that you'll see a lot of structures this term. In general, I'm not going to require you to memorize structures, but I will tell you when there's structures that I think that you should memorize. Okay? So we'll get to that. So Schrodinger had the idea that the roots of biology were not in tissues, they were not in organisms, they were not even in cells. Okay? The roots of biology are in molecules. And that dates to the 1930s. So that was only about 75 years ago that we really came to the realization that there's a molecular basis of biology, and it's from that molecular basis of biology that the term molecular biology comes from. Biochemistry is at the root of molecular biology because biochemicals are the molecules of molecular biology. Okay? Today we're immersed, absolutely swimming in information about molecular biology because upon that realization our focus turned from understanding macroscopic things about biology. Why is it that I have this birth defect? Why is it that I have blonde hair? Why is it that I have no hair? Okay? These have their roots in genes, in chromosomes. Understanding this was very, very important to us. And so when we turned our focus to that, by the 1950s, we understood exactly the structure of the DNA that gave rise or that held the information of the genes. By the 1970s, we began to see much more at the genetic level what was happening with those genes. And by the 1980s, we began developing some incredible tools that allowed us to unlock the secrets of the DNA. Those secrets are contained in the form of the sequence of bases that are there. We are in what people now describe as the post-genomic era. The post-genomic era refers to the fact that once we had enough ability to determine the sequence of bases for an entire genome, it created a new science called genomics. In 2001, the outlines and the essentially finished version of the human genome became available for the first time. And to give you an idea of that may seem to you like, well, okay, these are just little historical facts. But to give you an idea about how fast things have happened in biology, there's not a science on the face of the earth that has had the revolution that biology has had in the past 25 years. When I was a graduate student here at Oregon State, I got my PhD at Oregon State in 1986. When I was a student here at Oregon State, the thought that we would have the sequence, the DNA sequence of any genome, okay, was unfathomable. 1986, that was 23 years ago. Okay? One of the proposals for my PhD projects was that I determine the sequence of a human gene. And I said, no way, way too complex. Today, I could take any single one of you in this room and help you to clone that gene overnight. Okay? That's pretty phenomenal. We went from having very little ability to manipulate and understand DNA to being able to routinely determine sequences of millions of bases of DNA in a single day. That's what enabled the human genome sequence to become available. It took something like several years to determine the first genetic sequence of about 80 nucleotides, several years. Okay? The human genome, when we finally sat down and started doing it, took about 5, and today we can do a genome in probably a month. Okay? So there's been a revolution in terms of technology, there's been a revolution in terms of tools, and there's been a revolution in terms of our knowledge as well. Our knowledge is growing exponentially. Now the bad news for you guys is that means that there's a heck of a lot more you got to know than what I had to know when I went through. That's sort of a joke, but okay. But the reality is there's a lot more information there. What I want you to know are the highlights of that information, the important things that you're going to take away from this class for your own professions, for the things that you're going to be ultimately professionals in. Okay. Well, let's dive into this. The disease in the genome. As I said, there's no way today that you can pick up a newspaper and not see some medical advance every single day. I teach a class called Molecular Medicine. In that class, we have News of the Week. And we get inundated in that class with the medical news of the week, just the things that were reported that week. It's because we have what I would describe as the renaissance of biology. The renaissance being that we're uncovering, we have access to information like we've never had before, and we can start putting different pieces of information together and understand how all of these things are happening. What you see on the screen, of course, are chromosomes and representations of chromosomes, and chromosomes are at the root of information in biochemistry. We won't talk an awful lot about chromosomes this term. This term is more the sort of foundations of biochemistry. Next term in BB 451, we will, for the last half of the term, talk in considerable depth about molecular biology. But before we talk about molecular biology, we have to understand the sort of roots of biochemistry, the chemistry underlying all of that, in order to go forward. Okay? Evolutionary timeline. If we look at the evolutionary timeline, you'll see these in a variety of places. That evolutionary timeline puts us pretty late in the scheme, folks. We're not very long on this face of the earth, and hopefully we last half as long as the dinosaurs did. The tree of life. The tree of life breaks into three main branches, the three main branches being prokaryotic cells, eukaryotic cells, and Archaean cells. We, of course, are eukaryotes. Eukaryotes are organisms that have a nucleus. They have organelles like the nucleus, like mitochondria, like the ribosomes, the endoplasmic reticulum, and so forth. So that's what eukaryotic cells have. Prokaryotic cells, by contrast, are much simpler. They're what we refer to as the bacteria. And all prokaryotic cells are unicellular, meaning that they don't come together and clump together to make an organism. So all prokaryotic cells are unicellular. Some eukaryotic cells are unicellular as well. So we're an example of a multicellular eukaryotic organism. Yeast, on the other hand, are a unicellular eukaryotic organism. Yeast have organelles. They have nuclei. They have mitochondria. Okay? Archaean cells are the most recently discovered branch of the various kingdoms of life. And we won't really say much about them. I'll talk about them very briefly one point in this term, but they are thought to be a much more primitive form of life, and they're actually fairly hard to cultivate, and so we don't say a lot about them. I will say a lot this term, however, about the differences between prokaryotes and eukaryotes. So when I use those terms, I want you to keep those things in mind. Bacteria and higher cells. That's what we're talking about. DNA, everybody knows that A pairs with T, G pairs with C, etc. Okay? They're the bases. There's all the information. Okay? You have 7 billion base pairs of these. The base pairs, of course, A pairing with T, G pairing with C, as I said. Okay? And these come together in ways that allow information to be passed from one generation to the next. Again, we'll talk more in detail about that next term. I don't think I need to talk about that. Okay. Yeah, so that's the basics of what I want to say with respect to molecular biology. What I want to do now is turn our attention to some basic chemistry. A common question I get in this class is, do I need to have organic chemistry? The answer that I give is it's a prerequisite for the course, but if you're willing to waste your money on taking it without having it, I'm willing to take your money. Okay? Now, I say that, and I say that sort of jokingly, but I do let people in who haven't had organic chemistry. And in general, I don't recommend taking the course unless you've had organic chemistry. But I will also say I don't teach this as a chemistry form of biochemistry. There's a lot of ways of teaching biochemistry. And there are some people whose foundation in biochemistry is much more chemical in nature, and so everything they see is in terms of chemical reactions, et cetera, et cetera. We'll talk about chemical reactions this term. Okay? And we'll even talk about a few mechanisms this term. But for the most part, my roots are not in the chemistry side of things, but more on the molecular biological sides of things, the proteins, the DNA, the RNA, and so forth. That isn't to say that we shouldn't understand basics of chemistry. In freshman chemistry, you learn something, I hope, about hydrogen bonds. Hydrogen bonds arise from, hydrogen bonds, and by the way, the things I'm now talking about, yes, you are responsible for. Hydrogen bonds arise from unequal sharing of electrons. Okay? In freshman chemistry, you should have learned something about electronegativity. Electronegativity is a measure of the attraction of a nucleus for its electrons. That's the higher the electronegativity, the more strong the attraction of the nucleus for the electrons. If we compare the electronegativity of nitrogen with the electronegativity of hydrogen, what we discover is that nitrogen has a slightly higher electronegativity than does hydrogen, meaning that nitrogen pulls electrons towards itself more tightly than hydrogen pulls it towards itself. This pulls it towards itself. These are electrons that are being shared between nitrogen and hydrogen. One electron coming from hydrogen, one electron coming from nitrogen in this particular bond. If there's unequal sharing, that means those electrons are being pulled more closely towards nitrogen. It's not an ionization in which the electrons are completely given up, like we see with sodium chloride, where chloride takes the electron and sodium loses the electron. But instead, it's the big kid, little kid with respect to lunch money, right? Actually, the big kid, little kid with respect to lunch money is more like taking the electrons, isn't it? I take your lunch money and you're not going to have it back because I'm going to use it to eat with. Instead, the big kid, little kid is more like they're fighting over that lunch money, and the little kid isn't that much littler than the big kid. Bad analogy, but that's the best I can come up with on the top of my head. Okay? Oxygen has a greater electronegativity than hydrogen does. Now, as a result of these partial sharing of electrons, nitrogen gets the electrons closer to it, if you want to think about it that way, than hydrogen does. So nitrogen ends up being partially negative. That's what that little delta sign is there. Hydrogen ends up being partially positive. Well, these partial positives and partial negatives do, in fact, factor into electronegativity, because you can see in this case this partially positive hydrogen right here is attracted to this partially negative nitrogen over here, pulling them closer together. Plus attracts minus, right? It's actually this pulling between the partially positive hydrogen next to an oxygen in water that gives water the properties that it has. Water has a very high boiling point compared to other molecules of similar molecular weight. Okay? The molecular weight of water is 18. The molecular weight of carbon dioxide is 44. Yet at room temperature, water is a liquid, carbon dioxide is a gas. Why is that? Water is a liquid at room temperature because hydrogen bonds are literally holding these guys together in liquid form. Carbon dioxide doesn't have hydrogen bonds. Those forces that help stabilize water at room temperature aren't there in carbon dioxide. Carbon dioxide's out in the atmosphere. Okay? Those hydrogen bonds have a lot of force, and in water they're a very important force, as we're going to see, because those hydrogen bonds not only allow water to be liquid at room temperature, which is very important for us to be alive. Okay? Not only is that very important for us to be alive, but it's also very important for the molecules that are dissolved in water. We're going to see, when we talk about proteins, that NH bonds are very, very important bonds. And that NH bonds can make hydrogen bonds, both with themselves, like you see here, that is a hydrogen attracted to another nitrogen within a protein, but also between hydrogens on a nitrogen and, let's say, an oxygen in water. So proteins are soluble in water. Many proteins are soluble in water, not all of them, but many proteins are soluble in water because they can interact with water through hydrogen bonding. We're going to see when I talk about the structure of proteins that one of the most important forces in stabilizing a protein is the hydrogen bond. One of the most important forces in stabilizing a protein is the hydrogen bond. We're going to see structures in proteins for which hydrogen bonds are the most important structure holding them together. Okay? So hydrogen bonds are not something that we can easily dismiss. We need to think about that. There are terms that your book refers to with respect to donor and acceptor. A donor, a hydrogen bond, a bond donor is something that is, it's actually not labeled very well here, but a donor is actually this hydrogen right here. The acceptor is that which pulls that in that direction. Okay? I don't like those terms, so I won't use those terms, but that's what those refer to. Here's some examples of hydrogen bonds in biomolecules and some structures that we're going to see over and over in biomolecules. Okay? There's a carbonyl group. That oxygen right there is partially negative because that oxygen can pull electrons away from that carbon. There's a hydrogen that's partially positive. There's an interaction or an attraction that happens between them as a result of that. Okay? Here's an amine group that might be on an amino acid in a protein. There's a water, same sort of interaction, etc. If we swap things out, now we see that this carbonyl can actually form interactions with an amine on here. And we'll see that this interaction that happens right here is very, very important in the structure, in stabilizing the structures of proteins. For the first couple weeks of the course, you're going to hear me say an awful lot about structure, because structure is essential for function. You're going to hear that over and over. You're going to get tired of me saying that. Structure is important for function. The structure of a protein is critical for its function. If we destroy the structure of a protein, we destroy its function, period. Okay? Forces that stabilize structures of proteins are therefore very important to understand. We destabilize the structure of a protein through a variety of ways. Detergent will do it. One of the ways we kill bacteria on our hands, we wash our hands. Detergent, soap does that. The reason we cook our food, heat denatures proteins, and denaturing proteins in the bacteria and so forth in the food allow us to not be destroyed by the bacteria that's in there. And the reason that cooking and washing our hands and so forth works is because we are destroying the structure of proteins that are part of the bacteria that are there. Okay. Another consideration we'll have are van der Waals forces. And van der Waals forces are a little hard to understand. If there's only one thing that you take out of what I'm going to say about van der Waals forces here, it's this. And that is that van der Waals forces define for us an ideal distance of interaction. An ideal distance of interaction. If we look at trying to put two nuclei together, what we discover is that from a certain distance apart, there actually is an attraction. Okay? An attraction. The further down we are here, the more attractive, the stronger the attraction. So we start putting these nuclei together, we decide they get closer and closer and closer, and we discover, wow, they're getting more and more attracted to each other. It's kind of like singles dating or something, right? Yeah. That you're on the computer and here you're getting closer and closer to this person that you've been communicating with on the computer, and the attraction is getting stronger until you actually meet them. Okay? And then when you actually meet them, you get this sort of revulsion thinking, oh my God, what have I gotten myself into? How in the world did I ever think this? Get me out of here, please. And the repulsive forces take over at that point. Okay? Now, the repulsive forces, what this means is if you try to put two things together that don't want to go together, not unlike online dating, okay? You try to put two things together that don't want to go together, the repulsion, after a certain point, goes up to the power of 12 as the distance decreases. This repulsive force is enormous. It's why you can't put atoms too close to each other. And that's going to be a very important consideration for us when we look at the structure of proteins. Van der Waals interactions tell us that we can't put two nuclei too close together, because if we try to put them too close together, they're going to fight it with all their might, and I'm not going to be able to overcome that repulsive force. That repulsive force dictates how close together we can put atoms. And we start folding proteins, and we start putting atoms in close proximity that weren't in close proximity, there's going to be a limit how closely we can put them together. And that will determine things about how a protein folds. Okay, ice. There's ice. Memorize that structure for the exam. Okay? Hydrophobic effect. Okay? Now, I'm talking about some very general things here, and I will be honest with you, the first lecture is kind of chaotic because it's very much of an introduction. But I'm going to be talking about all of these things in some more detail as we go along. So I'm just kind of scratching the surface here. I guess what I'm trying to tell you up front is I'm not totally as disjointed as I am in the first lecture. I can be pretty disjointed, but I'm not as disjointed as I usually am in the first lecture. Hydrophobic forces. Hydrophobic forces are also important forces that are involved in determining protein structure. Hydrophobic forces. Hydrophobic forces you already know about a little bit. And what you know about hydrophobic forces are the fact that oil doesn't mix with water. Oil being hydrophobic tends to associate with itself. Water being hydrophilic, being more polar, tends to associate with itself, and never the twain shall meet. If you take a bottle of oil and vinegar, oil on the top, vinegar on the bottom, vinegar being an aqueous solution. And you shake it up. What happens? What do you see? You see droplets. It sort of mixes. It doesn't really mix, but you see little droplets. What did you just do? You applied energy, shaking, shaking, shaking. You're forcing something apart that doesn't want to come apart. And so they sort of mix. But what happens when you set that bottle down? It separates, right? Why does it separate? It separates because of hydrophobic interactions. Look at this up here. When I have those little droplets that I shook and shook and shook, what I did was I made those little droplets of oil, in this case, immersed in water, let's say, or vice versa. It doesn't really matter. When I do that, if this is oil and these are water molecules as it's drawn around here, these guys end up being extraordinarily organized. Nobody wants to go over next to this guy, but this thing's immersed in the middle of it. Okay? It's kind of like being, I guess, at a party and you've got one smelly person. Okay? And nobody wants to be around them, so everybody tries to get an equal distance away from them so that you don't want to be around that smelly person, right? Okay? You've probably been there. You organize yourself sort of as far away as you can get, and that's what these water droplets are doing. It becomes very organized. The second law of thermodynamics tells us that disorder is a force in the universe. Disorder is a force in the universe. It's a driving force if nothing else is there. There's no heat involved here. What brings these water droplets together is the fact that when we start putting water droplets together, we have less surface area per total amount of material. Therefore, we have less order. Disorder is driving this to the right because there's less order around. If we add up all the ones here that are ordered and all the ones here, and we compare it to over here, we'll see that we have more in this case than we have in this case. So by coming together, we're favoring disorder. We're favoring entropy. We're favoring the second law of thermodynamics. That's pretty cool. Okay? So hydrophobic forces are important forces for us to understand. Questions on that? Yes. Water is a polar molecule. Okay. Well, let's turn our attention then to thinking about not thermodynamics. And by the way, we do touch on thermodynamics, this term that'll happen later in the term. We're not going to focus on thermodynamics now, but we'll see later why that's important, because thermodynamics allows us to predict directions of reactions, and we're going to see how important that is later. But instead, I want to actually dive into some material that you probably think you know and you really don't. And that's because your freshman chemistry class really didn't teach you crap about chemistry. Okay? Didn't teach you crap about chemistry. And that's not your fault. Well, maybe in some cases it's your fault, but it's perhaps more a fault of the fact that we haven't really figured out practical ways to teach chemistry and mathematics. I'm a practical person in terms of chemistry and mathematical knowledge. I want things to have real meaning for you and not abstract meaning for you. And that's not always possible to do, but it's important to think about. Let's start with some definitions. You learned in freshman chemistry, if you didn't, you will now. pH is equal to the negative log of the hydrogen ion concentration. Log being a logarithm being something that anybody who's taking a senior class in science should know. Okay? Okay? That's what you should know. That's essential for you to know. On exams, I will not require you to memorize equations. I will give you every equation you need to know. There will not be one equation you'll need to memorize. You will need to manipulate that equation or those equations to show me your knowledge. You will not be able to use a calculator. Why? Because calculators allow you to manipulate equations, and you're the one that's going to manipulate the equation. Okay? Now, I'm not going to ask you what's the logarithm of 4, and you have to figure it out. That wouldn't be fair. But you will have to understand what logarithms have to do with this equation. Okay? Now, pH is the negative log of the hydrogen ion concentration. What does that mean? It means that as the hydrogen ion concentration increases, the pH decreases. Hydrogen ions are a measure of what's responsible for an acid. The more hydrogen ions we have present in a solution, the stronger the acid. Notice that is the concentration of hydrogen ions. Concentration measured as moles per liter, which is the same as molar, which is a capital M, which is not the same as moles, which is what I find half the people in this class don't know. Freshman chemistry failed you. You didn't fail freshman chemistry. Freshman chemistry failed you. How you like that turnaround, right? Hopefully biochemistry won't fail you either, but well, if that happens, you can come and you can blame me. Okay? Now, conversely, the POH. We don't hear as much about the POH, but the POH, by the same definition, is just the negative log of the OH minus concentration. As the hydroxide ion concentration goes down, goes up, the POH goes down. Okay? I could talk about the P Kevin Ahern, and if I talked about the P Kevin Ahern, I would hope that you would say, well, by logical extension, that would be the negative log of Kevin Ahern. And as the Kevin Ahern concentration goes up, the P Kevin Ahern goes down, right? Okay, so you start to see a pattern. We can therefore talk about pKa. pKa is known as the acid dissociation constant. And I always slur that when I try to say it too fast. The acid dissociation constant, okay, which is a measure of the strength of an acid. The pKa, which is the Ka is the acid dissociation constant. The pKa is the negative log of the Ka. Well, what is the Ka? The Ka is shown right here. Okay? If we look at acetic acid, for example, acetic acid, which I abbreviate as HAC, when we put it into water, it dissociates into protons, that's an H+, and AC-. The Ka then is the concentration of the proton times the concentration of AC- divided by the concentration of HAC. What does that tell us? It tells us, first of all, let me ask you guys, what does that tell us? What does that tell us about this acid when I put that up there on the board? Anybody? What does it tell us? What does the fact that I have this equation here tell me about what's happening in this reaction? I bet nobody can tell me. It tells you what? And what does that mean? Oh, okay, very good. Where are you? Good. Okay. It tells you how strong the acid is. We talk about in this class about strong acids and weak acids. Okay? For the most part, we're going to be talking about weak acids in the class. Strong acids aren't applicable to this equation. Do you know why? They completely ionize. Okay? So if I have HCl, when I put it in water, everything goes to H and everything goes to Cl-. When I have acetic acid and I put it in water, that's not what happens. To give you an idea, if I take 10,000 molecules of HAC, that's my starting material here, 10,000 molecules of HAC, and I put those 10,000 molecules of HAC into water, well, let's start up with HCl. If I put 10,000 molecules of HCl in water, I get 10,000 protons and 10,000 chloride ions. Completely dissociates. If I put 10,000 molecules of HAC into water, one of those will lose a proton, making one proton, one AC-, and 9,999 HACs waiting there to do something else. That's a weak acid. And as weak acids go, HAC is fairly strong. Okay? How do I measure the strength of a weak acid? I said it was fairly strong. How do you suppose I would measure the strength of a weak acid based on the things that I've already talked about? Nobody? No? Well, how much of it associates? Based on what I've talked about, how would you measure that? pKa. Okay? The lower the pKa, the higher the Ka. The higher the Ka, the stronger the acid. So the lower the pKa, the stronger a weak acid will be. If I look at the pKa for acetic acid, it's 4.76. Okay? If I look at the pKa for various groups on amino acids, I will see that they may be anywhere from 6 or 7 or 9 or 12. In some cases, they'll have a pKa of 2 or 4. I would certainly hope that if I asked you, if I gave you two acids and I said one acid has a pKa of 2 and one acid has a pKa of 4, you could tell me which one was the stronger of the two acids. One of the problems that I give you, it has exactly that sort of phenomenon there. Tell me why, based on these pKa's, that this is true. You'll see that in the problem sets. Now, you'll notice I haven't said a word about bases. I haven't said a word about conjugate acids, conjugate bases, or anything, and that's because I like keeping it simple. We're not going to talk about bases in this class. The one time we're going to use the word base is when we use strong base. Sodium hydroxide is a strong base. Based on what I said about strong acids, what would your definition, what happens with sodium hydroxide in water? It completely dissociates. We will not use the term base for something that accepts a proton. We will use the term salt. People really want to get into this class. You come in. Okay? We will use the term salt. All right? And the reason for that is when you start using the term base, you will get confused based on what your notion of what a base is. Okay? Now, I see that the old clock on the wall is saying it's time to cut short. Let us cut short, and I will see you guys on Wednesday.